Cholera is a life-threatening disease marked by severe diarrhea and is caused by the bacterium Vibrio cholerae. The disease is typically transmitted by the ingestion of food or water contaminated with human feces, easily allowing Vibrio cholerae into the intestinal tract. The cholera toxin has 6 subunits in 2 different types, A and B. Subunits are protein molecules that assemble with other protein molecules to form a protein complex. There is 1 copy of A subunit, which exhibits the toxin activity. There are 5 copies of B subunits that function in binding to host cell receptors and aid in bringing the A subunit into the cell.
The B subunits will bind onto a membrane lipid called the GM1 ganglioside. The A subunit has 2 peptide components.
An enzyme, protease, breaks down the disulfide bond between A1 and A2. The A2 reacts with the GM1 ganglioside, resulting in endocytosis and allowing the rest of the toxin to be transported into the endoplasmic reticulum. Once inside the ER, the A1 peptide detaches from the toxin. It then leaves the ER to the cytoplasm through the lumen.
The A1 peptide is an enzyme that catalyzes the addition of ADP ribose to the G protein, which keeps it in a constant on state. The active G protein binds to adenylate cyclase, which converts ATP to CAMP. This will remain constantly on making a load of CAMP. The CAMP in turn activates protein kinase A.
The protein kinase A will open up ion channels, such as the chloride channels. With the ions leaving the cells, it creates a concentration gradient. The cell will then diffuse the water out of the cell to keep an equilibrium. Since the G protein is constantly on, the process will never stop. With massive amounts of water leaving the cells, it causes diarrhea and severe dehydration. This ultimately leads to death. The feces then leads to the spread of cholera.